Hi, my name is Jordan Lancaster. I'm the Chief Executive Officer for Avery Therapeutics. First, I'd like to give a shout out to ARM. This is our first time being able to present at the Cell Gene meeting on the Mesa. We're very excited about this opportunity. Avery is working on some very cool things. This is a great platform for us to be able to share our mission with everybody. Avery is a company based here in Tucson, Arizona. Our technology was originally developed at the University of Arizona. And we are a platform technology. We're really working on ways of delivering therapeutic payload to those places that it needs to go. We're really focusing on, in the cardiovascular arena, particularly chronic heart failure of ischemic etiology. So these are going to be individuals that previously had a myocardial infarction, survived that event, but later on in life are developing heart failure. This is a very large unmet medical need. There's about 600,000 new cases diagnosed annually. And it's the #1 cause for hospital readmissions and deaths in the United States. So it's a very burdensome and expensive disease to treat. I want to make sure that you understand the path that this heart failure patient goes on once they've been diagnosed. Heart failure is a progressive disease. They will worsen over time. Their interaction with a clinician will start usually in about class 2 heart failure. They'll go in, shortness of breath, symptoms of fatigue with activity, and they will be managed on drug management. These drugs will make them feel better. They will treat the symptoms of the disease, but this is a short-lived phenomenon. Ultimately, this patient will be in and out of the hospital, worsening over time. The mortality rate for these patients is approaching about 50% over 5 years. As these patients progress, there are some things going on at the tissue and organ level, particularly an increase in volume of about 3x, and an increase in myocardial mass of about 3x. And so we can use these as we're looking at our preclinical data to mark how well we're able to impact the improvement that we're seeing with our therapeutic. And again, these parameters are correlating with that mortality and quality of life, increased volume and mass, worse mortality, and worse quality of life. So with Avery, what we want to do is interact with these patients as they're starting to not respond to that drug therapy. We want to do our implants using our myocardia tissue graft, which I'll introduce to you shortly. And really, at the end of the day, the outcome that we're looking for is improved quality of life. We want to make these patients feel better, and we want to keep them out of the hospital. So I'd like to introduce to you myocardia. This is our spontaneous and synchronously contracting cardiac tissue graft. This is really a new way of treating heart failure. This is our product. This is what a clinician will pick up out of a dish and implant directly onto the heart. That implant would look something like this, using a minimally invasive approach, such as a robotics or video-assisted VATS procedure. That clinician would first visually confirm the site of injury. They would second deploy the product directly to where it needs to go, broadly implanting onto the surface or epicardium of the heart, tacking on with a simple suturing technique. Before recovering that patient, that clinician is going to verify and visually confirm appropriate deployment of the product and confirm that it's placed in the appropriate area. That patient will be recovered and shortly sent home. So at Avery, we're really targeting to have an allogeneic cryopreserved product. This will allow us to have a cost-effective and scalable approach that will really be commercially adoptable. So I want to change gears and I want to dig into some of our data. I think it's really important for everybody to understand the impact and improvement that we've seen in our preclinical models. So I'm going to be sharing those with you now. So first, we're going to start with some of our rat data, a small animal model of heart failure. Top panel that you can see here is a rat that received an infarct. And what we're looking at here is the left ventricle. What we're looking at is a large transmural scar. That area that's showing blue is infarct, damaged tissue. This is going to be an animal with large infarct and a very poor LV function. This is going to be analogous to that class 3-4 patient. And so just to show you what the progression of heart failure looks like. Now, the bottom panel is a myocardia-treated animal. This is a different rat, same type of injury, same level of extent of damage. What you can see are some stark differences. First, the area that we implant the graft, that treated wall is much thicker. Second, you can see a change in composition as denoted by the red stain. Those are myocytes. Also, what you can make out from this slide are apparent changes in ventricular volume. The heart will dilate as a compensatory mechanism of heart failure. And what we're able to show, and you can clearly see this in the slide, is that after our graft treatment, those ventricles become smaller. We can measure those changes using echocardiography. I'm sharing those data on the right side of this slide. In systolic volume on the top, in diastolic volume on the bottom. And what you can really make out here is a bidirectional response. Our control animals are worsening over time. Those volumes are becoming larger as expected. But our myocardia-treated animals, those volumes are becoming smaller, suggesting that we'd be able to achieve decreased mortality and improved quality of life. So these are very exciting outcomes. Fast forward to a large animal model. We have a swine model of myocardial infarction. For this, an animal comes into our facility on a Friday. We do an MRI imaging modality. We can grade the size of injury that we create. You can see that on the left slide, denoted by the white, marked by the yellow arrows. This pig then comes back into our facility on a Monday, undergoes a procedure and a myocardial implant. That image is on the right. So 6 months after treatment, we're comparing the same animal. You can see here that we have a smaller infarcted region, as denoted by the yellow arrows, and also an apparent smaller ventricular volume. On the right side of this slide, again, I'm able to show you that in systolic and diastolic volumes that we're seeing after treatment. Again, we're seeing that very nice bidirectional response. Our control animals, as expected, those volumes are getting larger. Our myocardia animals, those volumes are becoming smaller. So again, an ideal outcome for this patient population. Now, we can take this a step farther, and I'm really excited to be able to present these data to you. Every animal that comes into our facility gets a special collar. That collar tracks their activity minute by minute for the full 6 months they're with us. So essentially, we're obtaining millions of data points that we can then quantify in a blinded manner to assess quality of life. Quality of life being the metric that we really want to impact for the heart failure patient population. On the left side of the slide, what we're doing is after that pig has had an infarct, been enrolled in the study, we're looking at what's happening with the control group and what's happening with our myocardia treated group. As you can see here, the control animals, they're not feeling well. They are less active. That's something that we would anticipate with heart failure. But our myocardia treated animals, we're seeing the opposite. We're seeing improvement in activity. These pigs are feeling better. Again, a very nice bidirectional response with our myocardia treatment. On the right side of the slide, we're looking at exercise activity. So we can subject our swine to prescribed activity events that are timed. And what we're able to show here is that during the course of study, our control animals are not able to recuperate any of their activity levels, but our myocardia treated animals are. So again, a bidirectional response with our myocardia treatment. Now, we're going to talk about mechanism of action. Our mechanism of action is going to be slightly different than anticipated. We're really going to be classifying ourselves as a non-integrating cell therapy technology. We're not expecting our cells today to persist for any extended period of time. The data that I've shared with you previously, those are all in immune competent animals where we have not provided any immunosuppressive agents. To distill down what we think the contributors of mechanism of action are really going to be the secretome. We've interrogated our myocardia product. We know that it has about 230 unique factors that are given off from it. Many of these are sequestered in a rich extracellular matrix network. Second of which is immune system. We know that our product generates an immune response. We think that immune response is going to be positive towards the outcome, similar to mesenchymal stem cell therapy. Third, the epicardium. Interestingly, the epicardium has come into light recently about being a signaling hub for the heart. The way that our product intimately interacts with the epicardium is something that we're looking into to see whether or not that helps drive the level of recovery that we've shared with you in our previous slides. So I want to talk to you a little bit about our product, about why it's unique. We've done a lot of design and development around the product to really make it as simple and straightforward as possible. Our product is stable and robust. We really wanted to eliminate any elements for shipping and equipment requirements at the operational site. So really our product will be delivered. It will be opened in the operating room and implanted. No special storage or reformulation required. Additionally, shelf life. Being able to cryopreserve our product is a game changer. We can manufacture our products in bulk. We can cryopreserve that product and then ship it around the world to where it needs to go. We've worked really hard to make our product physician friendly. We've got it in the hands of clinicians from day one. We've tested over in excess of a couple dozen biomaterials. And really the concept here is that we want that handling of the product to be as easy and straightforward as possible, that it doesn't tear, that it doesn't fold, that it doesn't bunch, and that it's easily deployable, especially in those minimally invasive procedures. Last is the easy deployment. We're not going to be requiring any extensive intraoperative imaging, any formulation requirements. Our package will seriously be opened up and product removed, product implanted. So we're really trying to make the product implantation as simple and straightforward as possible. Now, when we're looking at myocardia, we have a lot of advantages. We have a structurally robust tissue that's biologically potent. We've shared with you some of our extensive preclinical animal data. We also have a couple of manufacturing processes. We have open system manufacturing, but we've also spent a lot of time developing closed system manufacturing processes. In this process, our product would be manufactured within a bio bag. That product would then be cryopreserved in that bio bag, shipped in that bio bag, and then opened for the very first time in the operating room. This will allow us unprecedented scalability in addition to superb quality of control. Again, the ability to cryopreserve and reconstitute our product allows for long-term storage, worldwide distribution, and literally on-demand delivery. So Avery is a young company. We completed a Series A in 2019. We had an Interact meeting with the FDA in the first quarter of 2020. And we're going to be targeting our pre-IND by the end of 2020. Forward looking, we're going to be really driving our energy into getting into our phase 1 clinical trial and submitting for an RMAT application. So I've talked to you a lot about our cardiovascular program. We've talked a lot about chronic heart failure as our lead indication. We're also exploring non-ischemic dilated cardiomyopathy. In addition to arrhythmia, we have a lot of impressive preclinical data showing decrease in ventricular tachycardia in our swine and rat data. And we have an ongoing pilot study in swine to treat atrial fib, and we'll be happy to report those data as soon as they become available. Avery has an extensive intellectual property portfolio. Our original founding IP was based at the University of Arizona, and Avery has subsequently filed a number of additional patents that are really methods, composition, use, and manufacturing. And we're expanding worldwide prosecution of these patents in the countries that you can see on the left side of this slide. So with that, I'd really like to say thank you for everybody for the opportunity to be here. This is a great platform for us to be able to start telling our story to this great audience. You know, we're really excited to share MyCardia, which is really a really nice, robust, scalable product. It can be easily deployed. It's cryopreserved. We've got great proven efficacy and quality of life, a strong IP portfolio, and we're really here telling our story and also seeking partners that can help us accelerate our commercial success. With that, my contact information is on this slide, and I thank you again for the opportunity to be here.